Hello everyone, this is Juliana, and I want to welcome you back to the Exocrine Gland Biology podcast. This is an initiative of the Exocrine Gland Biology Group. I'm happy to be here with you for the fourth episode of the second season, where we are working on bringing senior and junior researchers together to discuss science.
Hi everyone, this is Bruno. Our podcast aims to expand the Exocrine Gland Biology community, and together with Juliana, I'm excited to introduce you today.
Today is a special episode. We have a bigger group than usual. We will be joined by two moderators, junior moderators, who will lead the conversation, and two guest speakers who have collaborated on impactful organoid research. Bruno, should we start by introducing our moderators, please?
Sure. Our moderators today are Ivan Lin and Henry Waith from Icahn School of Medicine in Mount Sinai in New York. Ivan and Henry, would you like to briefly introduce yourselves and your current positions and your main research focus?
Sure. I can start first. My name is Henry Wythe. I'm a fourth-year PhD student in Dr. Allison May's lab at the Icahn School of Medicine. We're really interested in understanding exocrine gland development, and my main research focuses on understanding the role of the immune system in regulating these processes of both salivary gland development and also tracheal submucosal gland development.
Hello, everyone. I'm Yvonne. I'm a third-year PhD student at Icahn School of Medicine in Mount Sinai, also under Allison May's lab. I also work on salivary gland development, particularly in how rare ion channel enriched cells contribute to these glandular development.
Thank you for your introduction. Now let's move to our guest speakers. We are honored to welcome Dr. Lin, who completed her postdoc training at the Utrecht University and recently started her own lab in the Shanghai Institute of Biochemistry and Cell Biology, Chinese Academy of Science. And Dr. Hans Clevers, professor of molecular genetics at the Utrecht University, guest science and advisor of the Organoid Research Group at the Princess Maxima Center, head of pharma research and early development, and also the director of Hosch Institutes of Human Biology. And Dr. Clevers has been recognized with numerous awards through his career and has published in high-impact journals, including Science, Nature Medicine, Nature Cancer, Cell, among others.
Today, we will focus on their collaborative paper that it's called The Human Airway Submucosal Gland Organoids to Study Respiratory Inflammation and Infection that was recently published in Cell Stem Cell. Dr. Lin is the first author of the paper. Dr. Clevers is the corresponding author and the senior author. So could you, each of you, introduce yourselves and share a little bit of your research interesting and give us a brief overview of the main objectives behind this study?
Maybe I shall start, although normally it's ladies first. But so yesterday I gave a talk for the university here in Utrecht in Holland, where I realized, I was introduced that actually it was 50 years ago that I started as a student in Utrecht. And then I spent some time in Boston, Harvard, learned molecular biology. This is the 80s, brought that back. I was an immunologist, and back in my own lab here, we wanted to find transcription factors that were unique to T cells and would explain how T cells are formed from the bone marrow. This led us to clone the TCF transcription factors for T cell factors that then turn out to be key components of the Wnt signaling pathway. And so I slowly my lab drifted away from being T cell immunologists to developmental biology, because Wnt signaling was then mostly Drosophila, C. elegans, frogs. So we basically had all these animals in the hospital where I was head of the clinical immunology department. Then the dean told me that was probably not the best place to do this research. And we moved to the current institute, Utrecht, already mentioned by Juliana, where Lin also did her postdoc. This is 2002 or so. Meanwhile, we had learned that the Wnt pathway was not just important during development, but actually was key also to drive stem cells in the adult body, particularly in the gut. That's where we concentrated our efforts then. We also had found with Bert Vogelstein that mutations in the Wnt pathway cause colon cancer. So we knew that Wnt was very important for gut stem cells being although not yet discovered, but being the most active stem cells of the mammalian body. And then if you activate the Wnt pathway, you get colon cancer. So we basically studied both and eventually found, this was Nick Barker, a postdoc who's now a professor in Singapore from England originally. We found a marker, LGR5, that uniquely marked tiny cells in the epithelium of the gut at the base of the crypts of Leberkuhn. And Nick could show that these LGR5 positive cells were the stem cells of the intestinal epithelium. He then found that LGR5 is a marker for many other stem cells and probably all epithelial stem cells, including the ones that Lin has been studying in the submucosal glands. And then 2 years later, 2009, Toshi Sato took up a project to try to culture these stem cells. And this was actually quite daring because people believed that normal cells cannot be cultured. And actually, I tried to convince a few postdocs. Nobody wanted to do this because they thought this will be the end of my career. I'll waste 3 years and then I'll have to leave and I will never be in academia again. But Toshi took it up. And we had learned quite a bit of the growth factor conditions in the crypts of Leberkuhn, in the gut epithelium, what actually keeps these stem cells so active there. This is, for instance, epithelial growth factor, but also Wnt. I already mentioned this, Wnt. And then we knew we had to block BMP. We do it in 3D. There was advice from Minna Bissell, who basically had pioneered Matrigel in 3D cultures of epithelia. And then much to our surprise, so we really wanted to take one stem cell and then make many stem cells, gut stem cells, but we got these structures. And then Toshi soon realized that they were sort of copies of the tissue they came from, the gut epithelium. So all the cell types of the gut epithelium were there, even the shapes were somewhat similar. And he called them mini guts, and we could grow them forever. That was also a surprise. You could transplant them, they make normal tissue, so they're not cancers. If you take a cell line, you will always get a tumor when you transplant, but these organoids, they're really normal tissue. And then CRISPR was available. With CRISPR, you can generate anything you want in these organoids. Lin might say a bit more about that. And then we learned, because LGR5 stem cells were not only in the gut, but there was LGR5 marked stem cells in many other organs, that slowly and steadily, if we modified the culture conditions a bit, usually make it a bit more complex, we could actually start growing other organs like stomach, I think was the first one, stomach epithelium. We did liver, we did pancreas, we did prostate, we did breast. Snake venom glands, actually reptilian venom glands can be grown as organoids. And so the model of the lab then, so this is the past 15 years, is essentially when a new postdoc joins the lab or a PhD student, we look, what tissues are still not studied by us, and then we find a clinic that can provide us the tissue, and then we start collaborating very closely with the clinicians there, maybe the scientists there. And then I guess Lin took up several of these projects, and one was the submucosal gland in the trachea that I had never heard of until Lin showed the results of her project to me. But that's it. That's sort of in a brief. And if I summarize, it's been, I think, very successful, this approach. So it's extremely opportunistic what we do. We were immunologists, developmental biologists, cancer biologists, gut biologists, and now we're biologists of any organ at the moment. But typically we depend on, so you have to have courage. You have to bring something to a new project, and we always bring organoid technology now. So we know how to create organoids. We don't know so much about the tissue that we want to study. Find good collaborators that actually will tell you, no, this is important. This is a question we might solve together, and that has worked really well. And I think Lin will tell us about a very good example of how this works.
Okay, maybe I will just get started. As Hans mentioned, so from 2020 to 2024, I was a postdoc working with Hans. So when working with organoids really shape how I think about human tissue biology, culturing the adult stem cell derived organoids in vitro, that the goal is to build an experimental system that are very dynamic because you capture all the cell heterogeneity in the adult tissues. And in a way, it's also very controllable. You can change the tissues, expose them to different cytokines, niche factors, and track how they become. And it's also a very nice traceable system. You can add, for instance, you can trace the cell identity by making a reporter of a typical cell types. And another way is also a very nice human biology system, because you can capture the human biology as close as possible in vitro. So when I joined Clever's lab, I besides the submucosal gland organoid project, I was initially working with gut organoids. I was trying to optimize the human intestine organoids. And in that project, we found that the cytokine IL-22 can induce the Paneth cells differentiation in the intestinal organoids. And later on, because my previous training background is with CRISPR, so we did a CRISPR screen with the organoid system. And we found transcription factors that are regulating the cell fate decision in the intestinal organoids. And then in the submucosal gland organoids paper is that I think it's really interesting paper, because as Hans says, we're trying to find something that we have never studied before in the lab. And I looked into literature, and I found that submucosal gland is a very important specialized secretory glands that are very specialized in human airway epithelial. And they have a very important role, but I think people always look into the alveoli, look into the lung biology, but airway biology probably less popular. So I was thinking maybe it's a good way to, learn something new, to study, to start a new project in the lab. So that's how I end up with the submucosal gland. Yeah.
And I guess one of our moderators knows a lot about submucosal gland.
Yeah, that was a very good match, actually. That's really cool.
Maybe I should add to what Lin said, that she mentioned that before she worked on the submucosal gland, she works on gut organoids, trying to improve that using interleukin-22. Interleukin-22 actually had been published in one of the major journals to be a driver of stem cells and of the growth of the epithelium, which we doubted. And I think Lin very beautifully showed that actually it doesn't do though. It doesn't drive the stem cells. And actually there's quite a few pharma companies that have been looking in IL-22 as a treatment for inflammatory bowel disease. And I think Lin showed that actually that will not help, because what you do, you make beautiful Paneth cells, which are one of the many different types of immune cells in the epithelium, but you do nothing to the stem cells. I think you probably destroyed a few projects in some pharma companies unintentionally.
That's good science, isn't it? Like that's how it should work. Not destroying the pharma, but like kind of complement each other. And another thing that I was curious about what Lin just described is that I know that you guys also have other projects that not just on the glands itself. Like there's a Nature Cons on the tongue that I read some time ago. Like how different it is for like go to the gut biology to the glands in terms of like organoids building. Is the pipeline like the same or talking like totally different?
Well, so I actually don't do any experiments, so I think Lin has to say how this works.
Yeah, so I think the main focus in the lab is definitely about the adult stem cell biology. In the lab, I think we do not do much about the embryonic stem cells or iPS cell derived organoids. It's really about adult stem cell derived. And I think there are a lot of things that share between the gastro, like the gut epithelial and the airway epithelial. They are both mucosal epithelial, right? They serve a very similar function, actually. They are, I like this metaphor that I think the mucosa tissue is like the wetland in the nature. Serve as a barrier function. It also provide habitat for the bacteria, for a virus, right? And meanwhile, it also has this important signaling transduction function. It sends what you breathe in will breathe out, and what you in the gut, it sends the nutrients that you uptake, and it transforms it into, for instance, the endocrine signals. That's one of the cell types I work with, the enteroendocrine cells. So I think they share a lot of similar things, and that means there definitely something similar in how to control the stem cells. They self-renew because it encounter a lot of environmental stimuli, right? You need to be actively respond to these challenge to divide and meanwhile differentiate at the same time. I think that's a very important concept we have when we try to build the organoids. We want to maintain the stem cells, maintain their identity in the cultures, but meanwhile, they still preserve the multipotency in the cultures, and they could give into all different kind of lineage in the system. I think that's something we share no matter what kind of tissue, adult tissue you are working with, but that's the core concept we have in the lab.
Yeah, but maybe Lin, because there are some general principles, I guess, for any tissue. Like typically, you need to activate the Wnt pathway, you need to activate the tyrosine kinase receptor, for instance, EGF or AGF. But what typically, whenever we sit down, postdoc and me or PhD student and me, and we come up with a new tissue, so then it takes like half a year or so a little bit longer to come up with a protocol where it actually is possible to grow. So how? Because I honestly don't know. What do you do in that half year? What happens?
That's a very nice question from you. So I think I would say most of the formulas are from the initial gut protocols we have established back in the days by Toshi, right? It's definitely about the like the very classic signaling pathway, how we manipulate them. But then when we look into individual tissues, for instance, the submucosal gland, I realized that actually they do secrete their own Wnt. So in the formulation, I will eventually remove those. For example, I remove the asbond3, I remove the Wnt surrogate. Then I compare different formulation and check whether these cells the stem cells are still there, right? They can still maintain their identity. And then I think that's how you do it. And eventually, for instance, there are people working on endometrium. Then you need hormones, which gut do not need. So you need to add something in. So you remove something, you add something in, and you compare them, and eventually you have a new formulation. But I guess all these formulas are not perfect. That's why we can optimize them, right? Like the human.
And where do you? Because I know that Toshi, when for the first, so there was only a 3-component, right? It was the Wnt amplifier R-spondin, and there was EGF, and there was a BMP inhibitor noggin. And that we knew from transgenic mouse experiments and knockout mice that actually these were the three. So inhibiting BMP, activating Wnt, activating EGF receptor. That should be maybe enough. And for mouse, it turned out to be enough, but for humans, we needed more. But then Toshi would go into look at what mutations you see in cancers of that particular tissue, because they say a little bit about what growth factors are needed or need to be blocked for cells to grow. So in your case, for the submucosal gland, do you have any and of course, like breast cancer, you think of estrogen, prostate, you think of testosterone, that's quite obvious. But sometimes there are totally non-obvious components in the growth factor cocktail that seem to be key.
I think for the airway organoids, we always have the FGF7 and FGF10, and these are two key growth factors we add actually from we have an AMBO paper, right, in the lab on airway organoids. So my formula was actually taken from there, and then I try to reduce the components there to make my organoids more have these states that the stem cell can proliferate, meanwhile differentiate. So sometimes in the lab we have protocol where we have the expansion stage and then we differentiate them. But for me, I always like to have just one formulation that the stem cells can have both stage at the same time. This is exactly what we have with the human intestinal organoids. During the differentiation protocols, they have these nice budding structures. We just need one formulation, and then we just culture them forever, just like the mouse organoids.
This would be a good maybe segue into like one of I think the first questions that me and Yvonne had kind of concerning this paper and this study. You've talked a lot about kind of what are the important signaling ligands and treatments you should use to maintain these cultures, and you've talked about comparing different tissues. You mentioned and acknowledge that you're not able to maintain this serous cell identity in the submucosal gland organoid structure, and you reference potential missing niche cues. I mean, this is obviously like a big question of how do we maintain these in culture, but what are your what do you suspect to be the missing links in kind of a more complete culture condition? What do you think are the most important kind of niche cues that aren't being utilized in a lot of different organoid cultures, or I guess in particular for this kind of submucosal gland culture?
Yeah, so in our SMG organoid system, we generate structures that are dominant by mucus producing cells. And these organoids, they capture key features of the mucus cell identity. For instance, they secrete MARC5B, right? It's a very specialized mucin expression, which is different from the surface airway mucus cells. So this suggests that the baseline epithelial differentiation program is still very well supported in vitro. But we do consistently observe that the serous cells are largely missing, or they only appear transiently after tissue isolation, and they fail to stabilize as a distinct population in these organoids. I think one important factor may be the cytokine signaling. Just like the partner cells that we cultured in the intestinal organoids, we can add IL-22, we have them. If we remove the IL-22, the organoids can still grow without the partner cells in these organoids. So in vivo, the serous cells are, I think they constantly exposed to a complex combination of cytokines and inflammatory mediators derived from immune cells or mesenchyme cells and the surrounding stroma. So many of these cytokines, they have multiple functions. They do not simply turn on the cell types, but instead they are maintaining, they are like a homeostasis cytokines. So in standard organoid cultures, we can still, this cytokine landscape is largely missing, I would say. So in our current protocols, we definitely can screen for these cytokines and see which could, derive these serous cells in vitro. And another factors I think is, I don't know, it could be a mechanical stimulation because there are highly active serous cell, secretory cell types that they normally experience repeated cycles of squeezing, like the pressure change in the airway. It's just like alveoli in the lung, right? They have this surface tension there to maintain their cell identity or cell type. So I think these mechanical forces could be another missing factors in vitro cultures. Yeah.
If we give another example, so Hugh Beumer, who was a PhD student in the lab and about the same time as Lin, but left for Basel and runs his own lab there. So he has continued to work on stomach organoids. And in stomach organoids, we could make all the cell types with one exception, the parietal cell, and that's the cell that produces the acid, secretes protons. And so we never saw a single, and we've tried many, many things. So what Joep tells me now is they can grow stomach organoids for long, or they can maintain them for a really long time in a sort of organoid on chip setting. And then after weeks and weeks of stable maintenance, they all of a sudden start seeing these parietal cells. So there it looks like it's just a matter of time and because we typically have rapidly growing organoids, or at least growing organoids. But if you now take them into a steady state, which is the normal situation of an organ, then apparently in the stomach you start seeing these parietal cells, which I had never fathomed that this could happen. And so maybe there would be another one. Just try, Lin, to keep them stable for months, and maybe they appear.
Yeah.
Cool. I guess a follow-up, have you considered things like age as well? I know you utilized kind of you got human donors to develop these organoids. If you get a cell source that's much younger, do you think there's also differences in the different cell types that you're able to derive or the types of organoids you're able to develop?
Regarding to the age of the organoids, I think we don't really have a choice of the age of the donors. But having said that, all the donors we have are adult tissues, derived. So I don't think I see a lot of difference, variability among these different donors. I think what you refer to maybe is more like like IPS or ES derived organoids. They might have a more fetal features. Yeah. So yeah, sorry. Your question is
If there are age differences, maybe I can say a little bit more. So not so much for the submucosal glands, but we have examples, quite good examples, where the age really matters. And one of them, for instance, is liver. So liver organoids, so unlike like gut organoids, for instance, which we can grow forever and they stay normal, liver organoids is essentially eventually telomeres get shorter of the chromosomes and they go into senescence. And the older the donor, the faster this happens. And so there, for instance, we have fetal organoids from human fetal tissue, which in Europe, at least in Holland, we are allowed to work with informed consents in place. That actually, so we have a number of fetal hepatocyte lines that we can grow forever, like the gut. And then we have some neonatal or say early pediatric samples that also grow quite well, but not as long. And then the older the donor, the shorter the span is. It's not so much what's in the organoids, but it's the lifespan is really different. And then we have another example, two examples actually, where tissues that we could never grow as organoids from adults. So one is brain, or from postnatal, and another is from pancreas. That actually, Dalila Hendricks and Benedetta Artigiani showed you can actually take human fetal brain organoids from a particular stage, and these can be propagated for a really long time and grow pretty fast. And it looks like we basically captured the tissue, we kept it in a state where normally the brain grows very fast during fetal development. And Amanda Anderson basically came up with something similar for pancreas, where she captured a mid-gestation stage from human fetal tissue, 14 weeks, 15 weeks, 16 weeks of gestation. If she grows pancreas organoids from these, they can make the entire pancreas. So they make the exocrine, interest of you guys. So there's the digestive enzymes like trypsin and lipases and things like that. They can also make the hormones, so they can make the endocrine components, and they make the ducts. And you can take a single cell and grow it back into an organoid line, and it can still make all those three. And it's almost like an ES cell, where an ES cell can do this for the entire body. These pancreas stem cells can do it for the different parts of the pancreas. And I'm sure that if we would work hard, we could do it for the lung as well, where we would get the precursor to alveoli, airways, and submucosal glands. But we have never, and apparently it's possible to take the cells out of a particular stage and then by the growth conditions, keep them in that state and then expand them for really long periods of time.
I
I
saw
love that. Yeah, sorry, Joanna, go ahead.
from one of your videos from before that you used to do the lavage to get the cells from the airway, but now is a biopsy. It's because of the stem cells. We need a different type of cell.
Lynn? Yeah.
Yeah, so, well, the submucosal glands, they do have a different subtype of stem cells compared to the airway basal cells, right? That's the myoepithelial cells. So I don't know how we get airway. I think it's how do we get biopsies before?
We did different things. We basically we at one point we thought it was interesting to grow organoids from without taking a biopsy from sputum. That works, and we did the same thing at about the same time for kidney and bladder organoids from urine. So if you capture the urine and you immediately cool it down and you make it isotonic, so you basically add a lot of buffer that the cells don't lyse, you can grow kidney, and then you get a zoo of organoids, the urethra, the bladder, the kidney, and then from airway, from lung, I think we only got airway organoids. So we never got submucosal. Well, we didn't really know that the submucosal glands existed then, I think. You explain that to us.
Yeah, but but having said that, so yeah, they do have a different population of stem cells. I don't think you can grow a submucosal gland organoids from the airway basal cells, right? Yeah.
That makes sense. When we're looking to the salivary glands, a lot of the differentiation that we're seeing, it's across like this kind of a transition area between the ducts and the acinar cells. Is it the same thing on the submucosal gland? So you need to have like that specific area, or you can grow with like wherever you're getting acinar cells or your myopithelial cells, wherever you're getting the tissue. Is it like my question is more is like a micro niche specific area that you need, or you just sort and throw everything there?
Oh, so we definitely need to micro dissect the gland out and then culture the gland as a whole in the culture as to make them form organoids. And then they do have a different, slightly different protocols with the surface airway organoids. And what we try is that we swap the cultures protocols. If we use the surface airway organoid media to culture the submucosal gland organoids, we can actually get them differentiate into CD80 cells, for instance, because it has been shown before in the mouse models that these myoepithelial cells can migrate out, differentiate into the airway basal cells or club cells, and to generate all different lineages in the surface airways. So that has been shown before, like these myoepithelial cells, they are the reserved stem cells for the basal for the surface airway. Yeah,
and
we
vice
actually
versa, the other way around. If you start from surface epithelium, you don't get them, right?
No, I don't get that. Yeah.
And do they grow slower than because that is often a complication. Do they grow slower than the surface airway?
Yes.
So
Yes.
then you have to be really careful to pick them out if you see them, because otherwise they get overgrown, right?
Sorry, you mean no?
So when you have a mix, when you see airway organoids coming up, you don't want them. So how do you keep your submucosal organoids stable?
When we separate the tissue, they are already in different wells. So I will not mix them.
Okay.
It's very It was pretty easy to identify what is a surface epithelium and what is a glandular structures when you isolate the tissues. Yeah.
And you never get mixed cultures because there's some contamination?
I don't think so.
Okay.
Because I handpicked those glands.
Yeah.
But it's close. I think this
great at dissecting the tissue, I guess. Yeah, I guess we always get surgical tissue, right? So these are typically older patients, maybe lung cancer patients.
Mm-hmm.
Yes.
Probably smokers, I would think.
Mm-hmm. I think on the note of like certain lung-related disease, I think one of the areas that people are very interested on, especially airway-related organoids, is also about chronic diseases, like for instance, like COPD, aging, fibrosis. There are pretty big burdens on the submucosal glands. So I have a question about, do you think like the current organoid system or the future organoid system can actually recapitulate like these long-term diseases, which are really hard to model in vivo? Or do you think there's already like certain strategies that could already help us kind of mimic certain like stages in those diseases as well?
What do you think, Lin?
Well, I think chronic disease like COPD, aging, fibrosis, I think it's also difficult to model them with a mouse model, right? It's essentially the physiology is different. You just really need to have a different way of model this chronicity. Organoid system, I think, in a way, you can have these repeated injury repair cycles or like a persistent inflammatory niche. It could actually change the stem cell behavior in the long term. And that could be a way to model this chronic disease. There's another thing I think would be to engineer the cellular age itself. But that it's probably more complicated, like use it towards like telomere manipulation or target this epigenetic editing to push the cells into a more age-like state. And then you can try to capture this late onset disease with the organoid system.
I can
Yeah,
add,
but I
yeah.
think, yeah, please go ahead.
I can add two thoughts. So for different chronic inflammatory bowel disease, our collaborators here in the Children's Hospital have shown that, say for Crohn's disease or colitis, if you make organoids from the diseased lesions, from the diseased areas in the gut, you actually, the first few passages, you can see they're heavily inflamed, but then when they're in culture for a while, they return to normal. And so implying that inflammatory bowel disease is not a primary problem of the epithelium, there's something else, the microbiome or the immune system, that basically constantly triggers and destroys epithelial cells and then puts the stem cells in overdrive. And then when you take the stem cells out and you turn them in organoids in an environment that they like, they lose that inflammatory signal. So that I think that's in part a challenge with these chronic inflammatory diseases, also like COPD or asthma, that actually we don't really know the cause, right? We know all sorts of interleukins that are up and down that contribute to the damage and all the regenerative efforts that are being made by the organ, but we don't really know what was the initiating cause of this disease. And as long as you don't know that, you can probably not really model the disease itself. You can model symptoms, and that's what we do in animals. I mean, animals. Many of these animal models have great symptoms, but they don't have the cause. And that's been a challenge, for my past years in Roger. I also saw this for COPD or for IBD or for SLE, for all of these chronic inflammatory or MS, is when you study this in the currently existing models, because they're not driven by the real cause, they're driven by other things, you will never find a cure. You'll find maybe symptomatic treatments, but you'll never find a cure for the disease. And that's I think, a challenge that maybe in animals is extremely hard or impossible to solve. And that's a challenge for, I think, the organoid field to really come up with. It's you probably have to add the real culprit, either the regulatory T cells or the microbiome, and then create situations where you can see the disease being initiated. Another thought, this is a bit of an hang-up of me. So fibrosis happens in any chronically damaged organ. No, it happens in the lungs, it happens in the liver, in the pancreas, in the gut. I'm strongly convinced that blocking fibrosis will not help the patient, because you're too late. Basically, fibrosis is a reaction, it's scar formation, a desperate attempt of the organ to keep its structure intact. And like when you have a myocardial infarction, your muscle is disappeared. There's no stem cells in the heart muscle. So basically the only rescue is now you replace that muscle by a scar, which is fibrotic. If you would block the formation of fibrosis in the heart, you die. Basically your heart would rupture. And so for liver and primary pulmonary fibrosis, I'm convinced that there's decades-long attempts to block fibrosis in an effort to help patients. I think that's not the target. The target is not the fibrosis. The fibrosis is a response of the patient. You really need to go after the cause, which is often unknown.
I think this is a really, I think, apt segue then into, I guess, also mentioned in the paper for kind of proposed future experiments is utilizing potential co-culture assays, particularly with the immune system, to better understand like those interactions, and particularly in the case of when you challenge with these different inflammatory molecules. And so I'm really interested in what you sort of envision with those co-cultures, because obviously the immune system is very, very diverse. There's lots of different cells. It's very tissue specific as well. I guess what cell types do you think would be most interesting to include in these co-cultures? And also kind of in the lens of understanding cause versus just symptom, like what do you expect to see? Do you even think that doing an immune system co-culture, do you think that would exacerbate kind of the effects that you see from these treatment with inflammatory molecules, or do you think it'd mitigate, it'd be a more kind of positive, kind of more tolerogenic response.
So in this study, so in this study, we use very defined cytokines like IL-1 beta, TNF, and IL-13 as like the minimum proxies for inflammation. And we already see that they behave different, right? The SMG and the surface epithelial. And we also have profiled a very specialized mucus cells response. Well, I guess the logical next step is not to throw in all immune cells at once, but to actually add complexity in a very tissue-specific manners, or like a disease context, depends on which disease you are working on. So I think for airway co-culture, I will start from the dominant resident compartments, or like disease-relevant compartments. For instance, the like the type 2 lymphocytes for allergic contexts, or for tissue-resident macrophage and neutrophils for the acute airway injury. So, and actually from the single cell data sets from human airway biopsies, they already tell us what kind of immune population actually sit next to the SMG or the SAEs in different disease contexts. So we can use that as a blueprint instead of just guessing. And then actually, I also think that in the future co-culture system, now with the engineering, the technology advance in the bioprinting, it would be nice to have a more defined way to co-culture. Like you have the glands sit in their compartments while immune cells are introduced more in like a vascular-like chamber or embedded in the matrix, where you can also have a very controlled exposure of cytokines into this whole system. And then over time, we can lay on additional specificity, not only the one type of immune cells, but many, many types. And we actually, I don't know whether we expect to see just one direction of either, exacerbate or mitigate. It could be, I'm actually more interested in how these cells communicate with each other. The immune cell communicate with epithelial cells, and how immune cells interact among themselves. So that's something as you say, is very complex system of the whole immune system in this interaction with epithelial cells.
Can I make a question on top of that? You're thinking and kind of creating a more complex system in this communication, but using like organoids, or even the thing that you do on your last figure of the paper, like sorting one cell and then co-culturing just the cells. Don't you think it kind of lacks a little bit the microenvironment, like the stromal cells? Is it possible to make like I know there's some papers out there, but how feasible it is to make like a co-culture of organoids itself, or like building the TME, like the microenvironment, and not just in one cell type? Because you just talk about like immune communication, and we know like the fibroblasts, they have a huge impact on the communication to the immune cells, or epithelial to the immune cells. So how do you see this? How do we build a microenvironment that's representative enough and it's not inside of a body?
Yeah, I think for the gut organoid, there are already paper published how you have all these different components, like as you say, the fibroblasts, epithelial cells, immune cells, actually autologous T cells from the same donor. You put them together. But for me, I think it's still too complex, because the readout is single cell RNA-seq, and that's probably the only readout you have. I think for now, I think it might be more reasonable to do it one by one layer by one layer, and the readout should be more controllable, traceable, and you should have a good, let's say, like how do you other than single cell RNA-seq, there should be other ways to ask the biological question. That's what I think.
Yeah, it's actually it's quite clear you've been in my lab, Lin, for a number of years. So my sense is of organoids, the strength is the simplicity, and of course it's really reductionist. But as soon as you add even one cell type, the numbers of controls you need to do, get horrendously. And then so people, I mean, you're not the first one to ask. I mean, that thousands of people have said this, but like stroma in cancer also, cancer tumor stroma is very important. I would always argue back, yeah, but actually when you add tumor stroma, you have no clue what you're actually adding, and every stroma sample will be different. We actually have replaced the stroma by the essential factors that the stroma makes, and we do it in an extremely controlled fashion. We know exactly how much EGF, how much wind, etc., etc. So it's a style of research which has served us well. So we basically focus on the most essential, simplest interaction. If you talk about two cells or something like that, try to see some ground rules. Typically, you get really black and white results. When you have a real phenomenon, it is usually anytime you do the experiment, you see it very strongly. So that means that you can actually rapidly move and make discoveries. And then we try to go back to the real situation and see if that rule that we have uncovered, if it holds in the complexity of a disease. Many other labs, basically, often with a lot of bioengineering, would basically try to experimentally bridge the two fields. But as Lin already said, with the current technology, it's exceedingly hard to not create. You'll have 25 96 well plates for one experiment to have all the controls over anything that you've added. So there's still enough. I mean, there's also very, very opportunistic. There's still enough questions to be asked that you can answer in a very simple system. So that's, I think, what I always try to discourage my lab to add complexity. Other labs do the opposite. So it's a style. It's a style of research.
I can
I
relate
think
to that because I was working with a bioprinter in my previous lab, and we were trying to print different types of cells together. And at the end, I decided to go simple. I was like, I'm just going to do one type of cell because then I can do something. It's very hard to establish a model when you have more than one type of cell. Even with a bioprinter there, even doing layer by layer was not something that you can just do. It's a big challenge. And I know, as you said, as a style, some labs are going to think that's the most important part, but some other labs are going to try something more simpler, and then they can add the factors. And I think that's very interesting to see that there is other types of views on that. I like it.
But actually, so the same people who would argue, you lack complexity with your organoids, they will be using cancer cell lines for their research where there is no complexity whatsoever. But that's fine, right? And then they transplant them to mice to make xenografts.
I think also on the note of like the more kind of like controlled, simplistic like ideas, I think there's one of the kind of like a debate that about the challenges that are very involved in like, standardizing how people are doing organoids because people have so many different approaches in a way. I think with Dr. Cleavers and Dr. Lin, all your expertise, do you think what certain steps do you think are very necessary to establish a good consistent method that could be used across different laboratories, because there are so many different methods of different papers all around, so it's been really interesting to see how to really establish these consistency.
I think I will give my naive insight, and later Hans can provide more insightful opinion. So I think organoid field are still very young. There are still many protocols are publishing every year from different labs, and they show that their system can give rise to more mature cell types. For instance, the liver organoids. This year we have two very good papers published on the adult hepatocyte organoids. But in terms of standardizing how you culture organoid, I think the most important thing is to standardize how you evaluate organoids. And it should be tissue specific. Like if, for instance, the gut organoids, how do you evaluate that this gut organoid is a good organoid, right? Do you have enough cell heterogeneity, and what kind of cell types you have in these organoids? Does it represent the region specificity of the biopsies, where the biopsy is from? So that's what I think. I think it is too early to talk about standardizing how you culture organoids.
Yeah. So speaking from my years of industry experience now, where industry has been quite slow to take up, particularly the 3D version of organoids for many reasons. It's complex. You need to be really skilled. You've worked with organoids. It's expensive, and it's not very reproducible. And so particularly if we want to see organoids, play a role in drug development or maybe in diagnostics, there need to be standardized protocols, standardized readouts, maybe automated versions of organoid growth, culture, and it has to be cheaper, it has to be simpler, it has to be more robust. So for that in particular, it's going to be key that standardized approaches and definitions of when do you call something an organoid. Now, having said that, I think in the IPS field, there's a lot more unpredictability in the outcome compared to the adult, because the adult stem cells that we start from, they're pretty dumb, right? They come from an organ, they know exactly what to do. And Lin, it's almost impossible to convince you your submucosal gland organoids will never become liver, right? There's no way to ever do that unless you knock out transcription factors and things like that. But so they don't have that problem. They are usually mature. They're very stable over time. You can continue to grow them. They're almost like a cell line in that sense. But still, if you grow them under different conditions, you'll get more of one cell type, or maybe you get more inflammation or something like that. So yeah, I would fully agree that standardization, particularly for more industrial applications, is going to be key. And there's quite a few activities there now. And I guess given that the FDA has now taken a very strong stance that we need to put much more emphasis on NAMs, on these, I don't even know what NAM stands for, but new something models.
Non-animal models, I think so.
Non-animal models, okay. So this is also AI-driven in silico models, organ on chip, organoids, et cetera. That one key development there would be is actually the standardization of any type of organoid that we use.
Do you feel that there's like a growth of people looking to your lab, like or looking for opportunities in your lab because of this? So we're seeing that UK and US, a lot of like countries are pushing a lot to not using animal models. And I was wondering, like, do you have like a growth of people looking for your lab to have like a postdoc or a PhD experience because of that, like a political decision?
Yeah, so I can tell you that my postdoc haven't had any hard time to find positions. And I often get asked, do you have anybody? And then the lab is extremely international. So, particularly now, we have actually quite a few people from China. I must say China has really embraced organoid technology. And I guess China is very strong in engineering. So I'm sure they'll be playing an important role in automation of the organoid approaches. Yeah, and then so what I saw in Roche also is where actually this predates me there, that actually pharmaceutical sciences there. So pharmaceutical sciences does all the pharmacology, safety, toxicity, dosing, metabolism of drugs. They had already been investing because of the then head of pharmacology in vitro human models. And there was a very active conversation, and more so now between the FDA and our pharmacologists over R is Roche over what are these organoids, what is the potential, what should happen. We now also see that the big companies that the CROs who support drug development with animal experiments or any other things doing assays, they now are getting more and more interested in organoids. So they're all building their own organoid capacity to either replace animals or to complement animal experimentation. So I think the whole drug development industry sees that this should be happening. Still, for many applications, it's not entirely clear if the assays exist at all and what it would take to, if they exist, what it would take to make them a standard assay.
Sure.
But I'm sure that with this push particularly from the FDA currently, this will move very rapidly in the next few years.
Yep. Amazing.
I don't want to be the boring person, but I have to wrap up. So before closing, that was an amazing discussion. It was very exciting to listen to you and learn from you. I just want to invite you, Dr. Cleavers and Dr. Lin, to give us a brief final reflection a takeaway from today's discussion or an advice for early career scientists, something that you hope our listeners could keep in mind moving forward, and then we can say goodbye.
Lynn, you want to give it a first try?
Oh, that's difficult. Well, I think I would just maybe give a little bit advice of being a junior group leader. Maybe I think in your audience there might be a lot of junior or ready to be to be a group leader postdoc. So I guess my first year as a group leader is honestly felt like the movie Everything Everywhere All at Once. So so there was suddenly like an overwhelming number of things competing for my attention, like setting up the lab, hiring and mentoring students, writing grants, teaching, reviewing papers, traveling for conference. So but meanwhile, I think it's nice to always interact with the people. Like, I like to talk to Hans, but he's very busy. But that encouraged me to think deeply and creatively about science, and that's very important. Yeah, so it's definitely a part of process of learning and how to move from doing experiments myself to enabling many experiments, ideas, and people to grow at the same time, building a team, shaping a scientific visions. So I really enjoy the conversations, and I think I definitely learned a lot today, and also from Hans. So yeah, work with organoids, yay.
Yeah, so I can add this. Actually, many of my PhDs and postdocs think that once they're a PI, they just sit in the office and people come in with their experiments and they write the papers and everything is easy. And then they find out when they are a PI that is like tenfold more complex or hundredfold more complex than running your project in the lab with a supportive PI. But you also discovered this now, Lynn. I understand. Yeah. So my wisdom or my advice would be, so I learned over the years without, I mean, I've never really thought much about how to, conduct science or how to be a PI or etc. But if I look back at what have worked for us in Utrecht, I think first of all, you cannot be somebody else's PI personality. You can only be, your personality is what you bring into your being a postdoc or being a PI. So you can look at other people, but you have to find your own way. And our way has been, I think we have always been very courageous. So we don't shy away from dumping into the deep in an area where we know nothing, but where we bring a technology. That's what we usually used to do. That's one thing I learned. Also, I tell my people always don't go into areas where there's already a lot of people or where there's already a lot of convictions on how things work and how you should think. Although you need to be courageous to jump into the dark. When you find something, you'll be the first, and you'll have some time to figure out exactly how it works. And another thing is I don't know if that worked for you, if you ever noticed, but I feel that so trust is extremely important. So trust in yourself, but trust in your colleagues. So and that comes with sharing. So basically, don't hide your results from your neighbor postdoc, or don't talk about your results before the paper is out. That all doesn't really help in the end. It doesn't make science fun if you want to be secretive and if you don't trust anybody you interact with. And I've rarely seen in my 40 years or so in academic science, I've very rarely seen that people have taken advantage of information or something that you've given them at your cost. But it pays itself back endlessly. So if you help somebody out now, and then 5 years later you might need that person for something else, and you can just read out, and people immediately reciprocate this. So that is so the combination of trust, sharing, and courage, I think has served us very well. Also has made it very enjoyable to be a scientist. I hope you you agree with what I say, Lynn. That is how
That's
you
great.
experience the lab. But
I learned from my previous experiences that collaboration is very important, and that we have to try to have good collaborations. As you're talking about different organs, we need to know different organs, so collaborations can help with that. Different technologies, collaboration can help with that too. So for me, collaboration is essential for research to move forward. And I really like the reflections you guys brought here, because that's how I see science too. So I want to thank you so much for joining us. And it was an honor to have you and learn from you, being able to discuss about science. I think that's the main objective of this podcast, is bringing people that can talk about science, talk about experiences, and then we can share with other researchers that maybe we cannot meet in person, but they can listen to us and they can learn from different people across the world. I think that's the main goal.
Thanks for having us. It was really nice.
I would also to thank you, Yvonne and Henry, for jumping with us in this conversation and with all the insights. Thank you so much for being here with us.
Yeah, thank you so much for having us. That was very enlightening. It was really fun.
Thank you so much.
To our listeners, thank you for spending this time with us. If you enjoyed today's podcast, don't forget to follow the Exocrine Gland Biology podcast on your favorite platform so you never miss what's coming next. We are on YouTube and Spotify, and we look forward to having you with us again for future episodes. Take care, and I'll see you next time. Bye.